Welcome to Pharma Toppers. Myself Piyush Agrawal, and today I'm going to discuss on glutamate as a neurotransmitter. So my flow of content will be like this: introduction, pathways and receptor, excitotoxicity, drugs, glycine. So L-glutamate is considered to be as an excitatory transmitter in the CNS, whereas GABA in the brain is considered to be as an inhibitory effect on the neuron, and glycine is found to be as an inhibitory transmitter in the spinal cord. So high concentration of the glutamate extracellularly and the calcium intracellularly are known to be neurotoxic. So these points are to be remembered. The widespread use of the glutamate as a taste enhancing food additive, and the Chinese restaurant syndrome as an acute attack of the neck stiffness and chest pain is well known. This all due to glutamate. So here you could see, friends, in this cycle the synthesis of the glutamate occurs through the glutamine under the influence of enzyme glutaminase, and also the TCA cycle products, that is alpha oxoglutarate, which also involved in the production of the glutamate, that is through the enzyme gamma T, that is GABA transaminase, which is an important enzyme in case of epileptic disorders, and the inhibitor of which is vigabatrin, used as a treatment. The synthesis of glycine takes place through the succinate, which is also one of the product of tricarboxylic acid cycles, through the glyoxylate under the influence of enzyme transaminase. And again the synthesis of GABA takes place through glutamate under the influence of enzyme glutamate aldehyde dehydrogenase, and thus as a result of which the GABA glutamate and glycine is synthesized. So here you could see, friends, the chart in which the receptors of glutamate has been explained, that is NMDA, AMPA, kainate and metabotropic. So the compositions: agonist, antagonist, modulators, calcium channel blockers, the mechanisms, locations and the functions has been explained. So this chart are important to be remembered. The main focus of this chart is basically the endogenous agonists, mainly the D-serine here, and again you could see the subunits, that is tetramer. This is also tetramer. This is also tetramer, but in case of metabotropic it is dimer and it is GPCR coupled. The type of subunits, that is NR1 and NR2, whereas in AMP it is glutamate receptor 1 to 4, whereas in case of kainate it is 5 to 7 and also kainate 1 and 2. So the other agonists, like here cycloserine, and the antagonists and the other modulators, basically the polyamines, magnesium and zinc in case of NMDA, whereas in case of AMP it is cyclothiazide, piracetam and eracetam. These are nootropic drugs. The channel blockers, the effector mechanism, that is ligand gated, which is mainly the slow kinetic and high calcium permeability, but in case of AMP it is fast kinetic and channel possesses the low calcium permeability, mainly through glutamate receptor type 2 subunit, and again in case of kainate it is fast kinetic and low calcium permeability, but in case of metabotropic it is GPCR, that is IP3 formation and release of calcium. The location here is of NMD and AMP is post-synaptic, whereas the kainate and metabotropic is pre and post-synaptic both, and the functions of which involve in nootropic NMD receptor is low EPSP, that is excitatory post-synaptic potential, synaptic plasticity involving the long-term potentiation and the long-term differentiation, basically related with the memory, and also the excitotoxicity. In case of AMP it is fast EPSP, in case of kainate it is presynaptic inhibition, and in case of metabotropic, which is also involving the excitotoxicity, basically the synaptic modulation. So here you see, the friends, the NMDA receptor basically the consist of the calcium channel blocking sites, channel blocking drugs, magnesium sites, glutamate sites where NMDA antagonists act, or the receptor site, the modulatory site, that is glycine antagonists, polyamine sites, and here the pore from where the sodium and calcium ion passes. And here you could see the subtype of excitatory amino acid transporter, that is 1, 2, 3, 4, 5, and they are the region in the brain. So here you could see, friends, the synapse which starts with the presynaptic neuron where the glutamate is synthesized, and it is taken up by the V-glut transporters, and which is released by excitotoxicities act on the metabotropic inotropic glutamate receptors and also through the excitatory amino acid receptors, with an exchange of 3 Na plus inside and 1 potassium outside and also 1 hydrogen ion inside, thus release and act on the postsynaptic neuron and enters into the glial cells. So here in glial cells the conversion of the glutamine occurs, whereas in case of presynaptic neuron the conversion of glutamine to glutamate occurs, and thus the response is achieved and the synaptic cycle is maintained. So here you could see, friends, this figure illustrates about the normal transmission and the conditioning transmission of the glutamate. Firstly, the NMDA channel is blocked by the magnesium ion, occurs at the physiological magnesium concentrations when the cell is normally polarized. But when a sustained polysynaptic depolarization produced by the glutamate acting repeatedly on the AMPA receptor activates and depolarizes the cell, however, removes the magnesium block, and NMDA receptor activation then allows the calcium to enter. So the calcium entry into the postsynaptic cell thereby activates the PKC and the phospholipase and the nitric oxide synthesis, resulting in the phosphorylation of the AMPA receptors initially during the induction phase, and is involved in the mechanism of the potentiation. Here you could see here, and later during the maintenance phase more AMPA is recruited to the postsynaptic membrane, as a result of which altered receptor trafficking and later still various other mediators or signaling pathways are activated, causing the structural change and leading to the permanent increase in the number of synaptic contacts. So the widespread use of glutamate as a taste enhancing food additive is already explained, and thereby this cycle has been caused the glutamate excitotoxicity if we take the increased amount of glutamate in the food, and thus the Chinese restaurant syndrome is causes. So here you could see, friends, the chart of the glutamate excitotoxicity, which is one of the part of neurodegenerative disorders, and as I've already explained, the high concentration of the glutamate extracellularly and the calcium intracellularly is considered to be as a neurotoxic. So I've explained about the first, second and the third site of this channel. So coming to the fourth site, that is depolarization, also opens the voltage gated calcium channels, releasing more glutamate. So metabotropic receptors cause the release of intracellular calcium from the sarcoplasmic reticulum. So thus the calcium sodium entry further contributes to the calcium entry by stimulating the calcium and sodium exchanger in case of site 5. So the depolarization inhibit or reverses the glutamate uptake in case of site 6, and thus increasing the extracellular glutamate concentrations. The mechanism that normally operates to counteract the rise in the intracellular calciums include calcium influx from the pump, that is the site 7 here basically. So again indirectly activating the sodium pump, that is from the site 8. So as a result of which the mitochondria and the endoplasmic reticulum, which act as a main sinker where the calcium is restored and normally keeps the intracellular calcium under control, but due to the excitotoxicity, what happens is the loading of the mitochondrial stores is increased, and as a result of which there is a disrupt in the mitochondrial function, and thus the ETC cycling is disrupted, and thus reducing the ATP synthesis and also the energy available for the membrane pump and for the calcium accumulation by the endoplasmic reticulum. Formation of the reactive oxygen species also enhanced, thus representing the danger point or the fatal at which the positive feedback mechanism exaggerates, and thus the process continues. So in this case, the free radical scavengers act as a protective mechanisms, and again the other drugs used here is glutamate release inhibitors, protease inhibitors, nitric oxide synthetase inhibitors, and mGlur antagonists are the main drugs to be used to control these glutamate excitotoxicity, as a result of which the neurodegenerative disorders is prevented. So coming to the next slides, the raised intracellular calcium level is neurotoxic because it causes the increase in the glutamate release, activation of the protease or calpains, which is a protein mainly involved in the necrosis, and also the lipase causing the membrane damage. Again, activation of the NOS, which produce the high concentration of nitric oxide in the presence of ROS, generate the peroxynitrate radicals and hydroxy-free radicals, which damages the biomolecules like DNA, enzyme, proteins, membrane lipids. In case of site 6, increase in the arachidonic acid release also plays a major role, which increases the free radical productions as well as inhibit the glutamate uptake. So these are the points to be remembered. The domoic acid, which is a glutamate analog, which is produced by the mucels, and the beta methyl aminoalanine present in the seeds of the local plant in the island of Guam, which is found to be the neurotoxic because it also enhances the glutamate. Riluzole, which is a compound that inhibits both the release as well as postsynaptic action of the glutamate. Memantine is found to be the NMD antagonist in the Alzheimer disorder. In case of domoic acid, a syndrome which combines the feature of dementia, paralysis, and Parkinson disorder, which is also been noticed or evidenced. So AMPA receptor antagonists produced overall CNS depressions include respiratory depressions, motor incoordinations with a little margin of safety. Positive AMPA modulators like cyclothiazide, which act by reducing the receptor desensitizations and thus improving the memory and cognitive performance, but it is found to be toxic. The other drugs like piracetam, which is a nootropic drug, or aniracetam, which are used in dementia, also sensitize the AMPA receptor. So blocking of this receptor prevents the depolarization of the postsynaptic cells and thus prevent the long-term potentiation which is involved in the memory, converting short-term into the long-term memory. So antagonists of these metabotropic receptors reduce the duration of LTP, and these agents are lipid soluble and thus crosses the blood-brain barrier. So LTP is reduced by agents that block the synthesis or the effects of nitric oxide or arachidonic acid. Anandamide, which is released by the postsynaptic cell, may also play a role by reducing the release of the GABA from inhibitory nerve endings. Ifenprodil and eliprodil blocks the polyamine actions, thus preventing the channel opening. So basically, concluding this slide, long-term potentiation and long-term depression plays a central role. The transmitter and the receptor which is involved is glutamate and NMD receptor in the learning and the memory. So here is some points on glycine, which is present in high concentration like 5 micromole per gram in gray matter of the spinal cord and produce the inhibitory effect in the spinal cord. Strychnine, a convulsant poison that acts mainly on the spinal cord and block the effect of synaptic inhibitory response in response to the glycine. Beta-alanine has a pharmacological effect and a pattern of distribution is very similar to those of the glycine, but its action is not blocked by the strychnine, which is important point to be remembered. The glycine receptor resembles the GABA receptor subtype, which is multimeric ionotropic receptor, mediated by the facilitation of the chloride channels, and is also associated with the muscle spasm and reflex hyperexcitability. Again, the toxin, that is tetanus toxin, which and also the bacterial toxin, which resembles the botulinum, act selectively to prevent the glycine release from the inhibitory neurons of the spinal cord, causing excessive reflex hyperexcitability and violent muscle spasm, and thereby the locked jaw disease is caused, and which is proved to be the fatal. So this are the charts. Just a brief synopsis where the transmitter involves is glutamate, glycine, and aspartate. The transporter blocker of glycine is sarcosine, which is important. The subtype receptors is mentioned. The agonist antagonist is also explained, and the receptor mechanism. And here is the different subtypes of AMPA, kinate, and NMDA. Agonists and actions is also explained here. So this chart is important to be remembered. So these are my references, And thank you. For more videos and materials or any queries, feel free to subscribe us at www.pharmatoppers.com.